Hello, my name is Gabriel Pope, and I will be observing alkaline phosphatase enzyme reactions under a spectrophotometer. So, in this lab, I will be analyzing the effects of alkaline phosphatase by looking at its absorbance. We will be using PMPP and BSA solutions in the cuvettes. A little bit of background on those solutions. BSA is meant to stabilize enzymes, and PMPP solutions are specifically used to identify alkaline phosphatase as a yellow color. The cuvettes that we'll be using will be labeled E1, E2, and we'll also have a negative control group called B or blank. And these cuvettes will be put under the spectrophotometer to see how much light is absorbed through these reactions. And enzymes, and background on enzymes as well. Enzymes are found in cells which catalyze reactions, and it breaks down certain materials or materials that we eat, like sugar, alcohol, or anything that we consume. And they also have to be a specific shape, and depending on the conditions of what the molecule is put in, or the protein is put in. And we will be using the spectrophotometer to use to analyze the absorbance, of course, like I said before. And I hypothesized that the reactions in E1 and E2 will be almost, if not identical, with the conditions that they are put in. So right here we have our E1, our figure legends. Here's the time 0 to 180 seconds. So for the most part, it's similar right here. I don't know. At 0 seconds in E1, the OD reading was very low. But other than that, the consistency and the growth is very similar when it comes to the absorbance. So the methods we use, the first step that we need to do is mess with our spectrophotometer and set it to live display absorbance with the wavelength of 410. Next, we get our 3 cuvettes and we label them, like I said before, E1, E2, and our negative control group B. And we will put 4.5 milliliters of the PMPP solution into each cuvette. Put 0.5 of the BSA solution into the B or blank cuvette, and we will mix the solutions with the parafilm on top of the tube. And we will do that by applying it on top, shaking it, putting it in there. And these reactions, never mind, we will zero out our spectrophotometer, blank the tube, and or put the blank tube in and measure the absorbance. And then for E1, we will put the 0.5 millimeters of the alkaline phosphatase that's in our water baths. That's another thing I forgot to add. Make sure that the enzymes are cold, because we're all about the conditions in this experiment. And we will put like I said before, we will put 0.5 milliliters of the enzyme into E1 and mix it just like we did with the blank solution. And we should also make sure that we're quick and that it's in the water bath. Put the E1 into the spectrophotometer, have your table, have your timers as well, and write down your OD reading every 30 seconds for 180 seconds. And also make sure to be quick. I can't stress that enough, because you need to be quick. And for E2, you repeat the same process as you do it with E1. So I don't know why it keeps doing this right here, but here's my graphs. There's only you can only see the slight differences, other than the major difference in 0 seconds of the graph but for the most part, the readings are pretty similar. And hold on. So in conclusion, before conducting this experiment with the enzymes, I knew what their function was because, of course, I took biology actually more than once in high school, and I learned about enzymes in both classes. But they are also crucial to human survival because of their ability to build muscle, remove toxins from the body, and break down and digest food, which we consume the nutrients to operate. And without the enzymes, we wouldn't have those fast reactions to provide us with the nutrients we need and operate with. And I didn't really realize how quick the enzymes would react to the solutions. Also, the OD readings, of course, like I said before, were very similar, and it supported my hypothesis. And the main priorities that I had during this experiment personally was trying to use the micropeptides properly, because I already knew that something was going to go wrong with the micropeptides. But that was my main priority, was making sure I got the correct amount of each solution and enzyme into the cuvettes. And I also had no previous knowledge before this experiment about the BSA and PMPP solutions, which I did some research about that after the experiment. But other than that, like I said before, there was only small differences in the readings. So in conclusion, there was only somewhat of a noticeable difference between the condition and the length of the reaction. And the limitation and assumptions I had were first being that I made the assumption that they would be identical. And second being is that the limitation I had was, of course, like I was referring to the micropeptide, I had to kind of redo my OD readings again. But other than that, the experiment was pretty smooth. And here are my references. And yeah, thank you for watching. Bye-bye.